Hello everyone, I would like to welcome you all in the second session of our today's event. My name is Ikra Sarfraz and I will be the moderator of this session, which will include talks from the fields of biochemistry, genetics, molecular biology, physiology and microbiology. Let's start this session with our first speaker, Dr. Haseeb Anwar from Government College University, Faisalabad, and he will be presenting his research on Bacillus perennis, a common ornamental plant with the potency of lowering glycemia and associated health biomarkers. Please, Dr. Haseeb Anwar.
Today I'm going to present in the third international conference on applied zoology, a virtual conference. I'm very thankful to the organizer for giving us the opportunity to present here in this forum. So the topic of my research is Bacillus perennis, common ornamental plant with a potency of lowering glycemia and associated health biomarkers. I am Dr. Haseeb Anwar from the Department of Physiology, Government College University, Faisalabad. So the outline of my presentation is about the pathogenesis of diabetes and the role of oxidative free radicals in the onset of diabetes, its aims and objectives, and the results which we obtained from this study. So about the pathogenesis of diabetes, there are two types of the pathogenesis. One is the genetic predisposition and the environmental factors. Collectively, both ultimately cause the decreased glucose uptake in the muscles, and it will lead to the hyperglycemia, and ultimately it will lead to the type 2 diabetes, which is actually due to the resistance of the insulin in the receptors at the targeted cells. If we talk about the role of the oxidative stress in the diabetes, due to the hyperglycemia, glycolysis cycle is disturbed, the mitochondrial metabolism is disturbed, the generation of free radicals, and the activation of few pathways involved in the stress signaling. Ultimately, the free radicals, they will cause DNA damage, lipid peroxidation, protein damage, and the stress signaling. Collectively, these will cause three major things. One is the insulin resistance, which leads to the type 2 diabetes mellitus, beta cell dysfunction, which leads to the diabetes type 1, and ultimately both of these types could lead to the diabetic complications. So this is a common daisy plant, family Asteraceae, and it has phenolic compounds mainly, and it has different beneficial health effects like anxiety, memory learning, and mainly it has anti-hyperlipidemic antioxidant effects, due to which we have selected this plant to test it in the management of diabetes mellitus. The objective is to evaluate the anti-hyperglycemic, antioxidant potential, immunomodulatory, hepatoprotective effects of this plant, flowers, ethanolic extract in the diabetic rats. If we talk about the experimental models, we have 5 groups comprising of 8 rats in each. One is the negative control without any treatment, without induction of diabetes. One is induced diabetes but without any treatment, and 3 groups comprised of the treatment. One of them is receiving the standard control drug, and the rest of the 2 groups will receive the plant ethanolic extract at 2 doses, 100 milligram and 200 milligram. We induce the diabetes in the rats by using the streptozotocin and nicotinamide injection, and the blood glucose level above the 200 milligrams per deciliter was considered as diabetic. So 3 days were taken for that, and we started the supplementation from the day 0, and the total experiment is comprised of 21 days. And at the end of the experiment, at 21st day, we actually decapitate the rats, and we collect the serum samples for the further analysis. These are the different analyses which we done. The anti-diabetic activity was comprised of 3 hormones: insulin, glucagon, and the glucose. For LFTs, different enzymes of the liver and albumin and total protein was counted. And to assess the oxidative stress, we did the total antioxidant capacity, total oxidant status, peroxidase, and vinyl esterase activity in the serum. For the immunomodulatory activity, we did the DNCB assay. About the results, it was seen that the glucagon hormone and the glucose level is significantly decreased in all the treated groups, including the standard control drug group and the ethanolic extract group in the glucagon and in the glucose results. However, in the insulin, the results were very significantly interesting, as the insulin level was significantly increased in the treated groups, particularly the standard control group and the 200 milligram ethanolic extract group, which was a good sign. And if we talk about the LFTs, the ALT and AST was significantly reduced in the ethanolic extract treated groups. However, the alkaline phosphatase is also significantly reduced. That reduction was in compared to the positive control and the standard drug control group. Albumin was also significantly decreased in all the groups as compared to the negative control, and the total protein, it was increased in the standard control group and the positive control group. If we talk about the oxidative stress, it was very interesting. It is just comprised of very interesting results. The total oxidant status was significantly reduced in all the treated groups as compared to the positive control. And similarly, total antioxidant capacity, it was significantly increased in the treated groups as compared to the positive control. However, the peroxidase and aryl esterase enzyme activity is also similar to the total antioxidant capacity activity. It was significantly increased in all the treated groups, which shows the good antioxidant effect of this plant ethanolic extract. If we talk about the immune modulatory activity was significantly increased in the 200 milligram ethanolic extract group at day 3 after this DNCB. So if we see the overall results, we can say that this plant extracts due to its phenolic contents, it reduces the oxidative stress, and ultimately it plays an antioxidative role, which helps to correct the hyperglycemia. It improves the beta cell function, it enhances the liver functions, and it also boosts the immune response of the body. Collectively, all of these effects were actually helpful, and you can say the responsible to reduce the oxidative stress, which ultimately in turn reduces the hyperglycemia by improving the beta cell function, and it also boosts the immune response and ultimately the liver function as well, which is a very good sign. And ethanolic extract, conclusively we can say that the ethanolic extract of this plant, AZ plant, plays a significant role in managing the diabetes mellitus by reducing the oxidative stress, and it also shows the hepatoprotective effect and the immune boosting effect in the hyperglycemic conditions, which was artificially induced in the rats. So thank you very much for your kind intention. I think this presentation will be very interesting and food for thought for all of you people. Thank you very much for kindly listening. I'm thankful.
Thank you, Dr. Hasib Anwar, for your nice presentation. And now I would like to invite the second speaker of this session, Nasibullah, from Cohort University of Science and Technology, and he will be talking about antibiotic susceptibility of bacteria causing subclinical mastitis in camel population of Southern Khyber Pakhtunkhwa. Please, Nasibullah.
Hello everyone. Welcome to the third International Conference on Applied Zoology 2020, organized by the Applied Zoological Society of Pakistan and Quaid-e-Azam University, Islamabad, Pakistan. My name is Nasibullah from Department of Zoology, Cohort University of Science and Technology, Cohort. I completed my research work under the supervision of Dr. Kalimullah on the topic antibiotic susceptibility of bacteria causing subclinical mastitis in camel population of southern Khyber Pakhtunkhwa, Pakistan. Introduction. Mastitis is an inflammatory disease of mammary gland of lactating animal, including camels, causing decrease in the milk production and leading to huge economic losses. There are two types of mastitis, clinical and subclinical. Non-observable form of mastitis such as no visible abnormality of either the milk or the udder is known as subclinical mastitis. Subclinical mastitis is a real threat to dairy farmers as well as for the milk consumer. There are different etiological agents of mastitis, but Staphylococcus aureus is the most common causative agent of mastitis worldwide. Objectives. The first objective of my research work was to find out the prevalence of mastitis in the camel population of southern Khyber Pakhtunkhwa. The second objective was to determine the antibiotic sensitivity profile of bacteria causing mastitis in camel of the study area. Material and methods. Study area and study population. Milk samples were collected from camel population in three districts, Lakimarwat, Diehan and Trang. Milk sample collection. During sample collection, the udder of camel was washed with water and disinfected with 70% ethanol. 10 ml of milk sample was collected in aseptic plain tube and were placed in the ice box for 4 degrees centigrade for further analysis. Screening for mastitis. Collected milk samples were tested for mastitis using surf field mastitis test. Identification of bacteria. The bacteria were isolated by multiple streaking and were characterized by chemically using coagulase and catalase tests. Antibiotic sensitivity test for bacteria. An antibiogram assay were performed using different antibiotics against bacteria that were causing mastitis in the camel. Statistical analysis. Data obtained were analyzed through SPSS. Results. This graph shows the overall prevalence of multiple drug resistance, that is SRS, from the total milk sample. A total of 87 milk samples were taken from the camel, in which 28 milk samples were
potential of Agonia indica from Cholistan desert against clinical multi-drug resistant bacteria. Pakistan is one of the richest countries in medicinal flora. Cholistan desert covered the 2.6 million hectares of Pakistan. Fagonia indica present in subtropical and hot temperature regions like Morocco, Tunisia, Algeria, Cyprus, Saudi Arabia, and dry regions of salty rocks of India and western Pakistan. It is mostly used in the Indo-Pak for the treatment of fever, urinary discharges, asthma, liver troubles, tumors, and peptic ulcer diseases. Antimicrobial activity of Fagonia indica has been reported against different microbes like Salmonella typhi, Escherichia coli, and Staphylococcus epidermidis. So, keeping in view the threat of multi-drug resistant bacteria, present study has been designed to investigate the in vitro antibacterial activity of different extracts of Fagonia indica prepared by different extraction methods against multi-drug resistant bacteria. Effect of Fagonia indica on biofilm formation and adhesion was also evaluated in our study. First of all, Fagonia indica plant was collected randomly from Tehsil Yazman, Cholistan district, Bahawalpur, Punjab, Pakistan, and after the identification, the collected plants were rinsed with distilled water and finally dried in shade. Dried materials, that is the roots and aerial parts, were ground in a mixture grinder and into fine powder. Powder material of each plant part was sequentially extracted by soft flat apparatus using different solvents in increasing polarity. After plant collection, the antibacterial activity of plant extract was evaluated against 4 multi-drug resistant clinical isolates, that is Staphylococcus aureus, Pseudomonas, aeruginosa, Escherichia coli, and Klebsiella pneumonia. After this inoculum preparation, each bacterial strain was subcultured in algae broth at 37 degrees centigrade. The growth of bacteria was harvested. Antibacterial activity of different plant extracts was determined using the agar-agar diffusion method. Next, the minimum inhibitory concentration and minimum bacterial concentration of plant extract was determined by broth microdilution method DMSO was used as a negative control and ampicillin was used as positive control. Detection of cellular protein leakage. In order to detect the disintegration of cell envelope after the treatment of cells with plant extract exhibiting the lowest minimum inhibitory cellular protein leakage was measured. The absorbance of the suspensions was measured at 595 nanometer. Then the cell attachment assay was used to determine the effect of plant extract on adhesion of bacteria to polystyrene surfaces. Negative and positive control were also included in each experiment. Biofilm formation was used to assess the effect of plant extracts on biofilm formation of target bacteria. Finally, the absorbance of the eluted suspensions was measured at 546 nanometer. All the experiments were performed in triplicate. Now, the highest yields were found for chloroform extract of Phagonia made from roots followed by methanolic extract obtained from aerial parts. Our revealed that Phagonia extract has exhibited antibacterial activity in a dose-dependent manner since increasing the concentration of extract increase the diameter zone of inhibition. Diameter of zone of inhibition range from 5 to 15 millimeter and the largest zone of inhibition was observed against Staphylococcus aureus using chloroform extract of plant made from results also indicate that extract made from aerial parts of Phagonia indica were more effective against tested bacteria as compared to the extract made from roots. Next, the results indicated that minimum quantify biofilm formation in the presence and absence of sub-inhibitory concentration of chloroform extract of Phagonia. This data also indicated that biofilm forming capacity was increased in a time-dependent manner among multi-drug resistant bacteria and maximum capacity was observed for Pseudomonas aeruginosa followed by Klebsiella pneumonia. It was also observed that sub-inhibitory concentration of chloroform extract of Phagonia significantly reduced the biofilm forming capacity of tested bacteria, and percentage inhibition of biofilm formation was found to be 30.33 to 71.84% as compared with control cells. Our data also revealed that after 48 hours of incubation, greater inhibition was observed for Staphylococcus aureus, that is 71.84%, followed by Pseudomonas pneumonia. Our study clearly suggested that efficient role of Phagonia indica extract as anti-adhesion and anti-biofilm agents against multi-drug resistant bacteria. Biofilm formation is considered to be a major public health concern since more than 80% of microbial infections of human beings involve biofilm formers. Therefore, current studies significant mentioning the role of Phagonia collected from Cholistan Desert biofilm inhibitors. However, in future, there is need to identify individual compounds from this plant responsible for this inhibition and also their molecular mechanism of action against antibiotic resistant bacteria. Thank you.
Thank you, Dr. Nosheen Aslam, for an informative talk. And now I will move towards Maham Fatma from University of Agriculture, Faisalabad. And she will be talking about antibiogram and biofilm formation potential of Pseudomonas aeruginosa.
Assalamu alaikum. My name is Maham Fatma. I am from Faisalabad. I have completed my graduation in microbiology from Agriculture University of Faisalabad. Now I have completed my MPhil from the same university, University of Agriculture, Faisalabad. Now my research topic is antibiogram and biofilm aeruginosa isolated from hospital environment. Firstly, I discussed about introduction. What is Pseudomonas aeruginosa? Pseudomonas aeruginosa firstly isolated in 1882 by visit from green pus. It is ubiquitous in nature, which has a tremendous capability to evolve and growing during a sort of environment conditions like clinics. It can first of all sample collection. Total 40 samples were collected from hospital environment like tap water, sink, catheter, floor, etc. Samples were collected with the help of a sterile swabs. Then फिर आ जाता है swabbing. Swabbing was done on McConkey agar and incubated for 1 day at 37 degrees centigrade. Then morphology. Morphology observes the characteristics of isolated bacteria was done by gram staining. Then further purification process. For purification was done on McConkey agar. Now, confirmatory test. Confirmatory test was performed in which catalase, oxidase, methylate citrate or indole test was performed. Then antibiotic susceptibility testing. The antibiotic susceptibility testing was done on monomolar Hinton agar through disc diffusion method. Then perform biofilm formation test through microtiter plate method. And now these are results. These are you can see the picture of Pseudomonas aeruginosa shows colorless colonies on McConkey agar. Colorless colonies on McConkey agar. You can see, and under microscope it is gram-negative bacteria, rod-shaped bacteria at 100x on light microscope. For biochemical test for Pseudomonas aeruginosa for confirmation, first oxidase test is positive for Pseudomonas aeruginosa, catalase test is positive for Pseudomonas aeruginosa, then citrate utilization test is positive for Pseudomonas aeruginosa, and indole and methylate test are negative for Pseudomonas aeruginosa. You can see results. And for confirmation, out of 40, 15 isolates who are Pseudomonas aeruginosa who are Pseudomonas aeruginosa, and you can see the occurrence of Pseudomonas aeruginosa isolated in hospital environment. You can see this figure, positive and 15 samples, positive samples. You can see table out of 40. Then I perform antibiotic sensitivity test result. By disc diffusion method, you can see the zone of inhibition of Pseudomonas aeruginosa isolated from hospital environment. Sample gaze for multiple drugs. Multiple drugs in which gentamicin, meropenem, and tetracycline, piperacin, or ciprofloxacin used. In which isolated bacteria greatest showed resistance against tetracycline and least showed resistance to the piperacillin. You can see table. Then biofilm quantification. By following the OD values, the production of biofilm isolation was classified into 3 groups: weak, moderate, and strong producers, in which 9 isolates were strong, showed strong biofilm formation, and 4 isolates moderate biofilm, and 2 isolates showed weak biofilm formation. You can see OD values of biofilm formation. According to OD values, we classified into weak, moderate, or strong biofilm formation. And you can see figure degree of biofilm formation. You can see figure. At the end, conclusion. In my conclusion, environment in hospitals are the main vehicle of Pseudomonas species, and therefore the patient and the people who are working in this area may must attention to their personal hygiene in order to avoid Pseudomonas infection. Antibiotic susceptibility testing is done to help to choose the antibiotics that will be the most effective against Pseudomonas infection. Multi-drug resistant of Pseudomonas aeruginosa is a major issue in the nosocomial infection, and piperacillin, in my research work, piperacillin may be effective in treating Pseudomonas infections, and strong biofilm producing isolate may be one of the factors leading to antibiotic resistance to a broad range of antibiotics. These are references, and thank you for your attention.
Thank you, Maham Fatma, for presenting your research at ICAS 2020. And now it's turn for Shama Ehsan from Government College University, Faisalabad. And she will present her research on antifungal potential of selected plant extracts against ochratoxin producing Penicillium verrucosum. Please, Shama Ehsan.
Assalamu alaikum, all of you. My name is Shama Ehsan. I'm here to present my research work. My topic is antifungal potential of selected plant extracts against the ochratoxin producing Penicillium verrucosum. Now move towards the introduction. As we all know, poultry is one of the fastest growing segments of the agriculture sectors, and feed play an important role in the success of poultry production. Poultry diets are formulated from a mixture of ingredients that include cereal grain, plant protein sources, animal byproducts, vitamin and mineral supplements, and food additives. As since it is used throughout the year, so it is stored for many long times, for months, years, and many factors play important role in fungal growth that contaminate the feed. That is feed composition, prolonged storage, temperature, moisture. Fungal proliferates after getting favorable condition and secrete mycotoxins. Most of the mycotoxins that are considered very important are produced by three genera of fungi: Aspergillus, Penicillium, and Fusarium. Penicillium is an important ochratoxin producer that produce ochratoxin A in cereal and cereal products. Ochratoxin is nephrotoxic, teratogenic, and immunotoxic. Primary target of ochratoxin is kidney. Sign of ochratoxin toxicity in poultry include weakness, decreased feed consumption, reduced growth rate, reduced egg production, and excessive mortality. Then I discuss about detoxification of ochratoxin that can be done by chemical, physical, and biological method. But in physical method, we use heat treatment, radiations, then sorting, separation. In chemical methods, we use different chemicals, but in chemicals have toxic effect that cause carcinogenity, teratogenity, and hormonal imbalance. So this method is not safe. But biological methods are more effective and safe for ochratoxin radiation, and plants are the rich source of in this regard we use plants, and plant are the rich source of phytochemicals. In my study I use 4 plants, and these plants produce many substances. Ocimum basilicum and Eucalyptus globulus have bioactive compounds like alkaloids, tannins, flavonoids, and phenolic compounds. These metabolites have antifungal properties. Objectives: isolation and identification of ochratoxin producing Penicillium verrucosum from poultry feed. Termination of the antifungal potential of selected plants extracts against Penicillium verrucosum. Now discuss about material and methods. I use 4 poultry feed samples that are collected from 4 different localities of Faisalabad. Then I isolated Penicillium verrucosum on 2 media. First one is yeast extract screws glycerol agar, and second is malt extract agar. Then identify Panthelium varicosum macroscopic and microscopically. Macroscopically, I see the shape, texture, and growing zone on media. And microscopically, I use lactophenol cotton blue stain. In my study, I used four plants: Eucalyptus globulus, Osima basilicum, Medicago sativa, and Aloe vera. These plants I collected and checked their antifungal potential. Then plant extracts by Soxhlet apparatus. And first I collect the plant, then shade it, dry it, then grind it, then make thimble and make ethanolic and methanolic extracts, and then concentrated by rotary evaporated, and then solidified. Antifungal activity and minimum inhibiting concentrations are determined by diffusion methods. That is, I use agar-well diffusion method. And now I discuss about results. This is the mixed culture. It is the result of spreading. I spread feed on growth media. This is yeast success grows glycerol agars, and different microbiota are produced in this plate, Aspergillus, Penicillium, and many other fungi are also present, but bacteria is not present because I use chloramphenicol as antibacterial. Then I isolated pure culture of Panthelium varicosum. Then I grow these Panthelium varicosums on malt extract agar. Then identify the Panthelium varicosums microscopically and results of antifungal assay. I use 4 plants and ethanolic extracts of Eucalyptus globulus and Osima basilicum showed positive result in antifungal assay, while ethanolic extracts of Medicago sativa and methanolic extracts of aloe vera showed negative effect against Panthelium varicosum. This is the result of methanolic extracts of aloe vera that showed no inhibitive effect against Panthelium varicosum. Minimum inhibitory concentrations. Ethanolic extracts of Eucalyptus globulus showed inhibitory effect at 10 mg per ml minimum concentrations. And ethanolic extract of Osima basilicum showed inhibitory effect at 20 mg per ml minimum concentration. Then I determine zones of their antifungal activity. Eucalyptus globulus show average zone 9.5, and the positive control. In positive control, I use itracanazoles as antifungal that give average 11 diameter zone. In negative control, I use ethanol. Osima basilicum average diameter is 7.5, and the positive control same 11, and negative control is 0. I concluded that ethanolic extracts of Eucalyptus globulus and Osima basilicum have potential to kill ochratoxin producing Panthelium species. Therefore, it can be used for subsequent addition in poultry feed. That's all. Thank you for your attention.
Thank you, Shamaya-san, for sharing your research with us. And now the next speaker of this session is Muhammad Asim from University of Agriculture, Faisalabad, and he will present his research on the role of mammalian target of rapamycin mTOR in aging. Please, Muhammad Asim.
Bismillahirrahmanirrahim. My name is Muhammad Asim. I am PhD scholar in the Institute of Animal and Dairy Sciences in the University of Agriculture, Faisalabad. I am presenting the role of mammalian target of rapamycin in aging. First, we define the aging. Aging is a time-dependent physiological functional decline that affects most living organisms, supported by alterations of several molecular pathways, and represents the most profound risk factor for many non-communicable diseases. For a long time, it was a debate either aging is a disease. To answer, Blagoskolny defined aging as a disease on the basis of potentially available specific treatments. Similarly, Delman described the aging process as a normal disease. So the sum of all age-related diseases or a key pre-disease is known as aging. Aging is a primary risk factor for major human pathologies, including cancer, diabetes, cardiovascular disorders, and neurodegenerative diseases. What are the common denominators of the aging? Several cellular processes which represents common denominators of the aging in different organisms are genomic instability, telomere attraction, epigenetic alterations, loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, cellular senses, stem cell exhaustion, altered intracellular communications. The mammalian target of rapamycin plays a pivotal role in the aging. Mammalian target of rapamycin, mTOR, is actually a serine threonine kinase belong to phosphoinositide kinase related family. It's a functional enzyme found within two distinct complexes, which is mTOR complex 1 and mTOR complex 2, both characterized by different protein partners and specific substrates. mTOR complex 1 activated by several growth factors through phosphoinositide 3 kinase related kinases family and AKT kinase signaling. mTOR complex 1 is also activated by nutrients like amino acids and phosphates and repressed by AMP activated protein kinase AMPK, a key sensor of cellular energy status. It's involved in cell growth, proliferation, survival, motility, autophagy, and protein synthesis. Low level of insulin and insulin growth factor 1, two growth factors actively, the two growth factors which are low levels of insulin and insulin growth factor 1 are the two growth factors which activated mTOR and induced by the caloric restriction and associated with an health span improved and longevity increase. mTOR complex 2 is less sensitive as compared to the mTOR complex 1 to the rapamycin and inhibited by the chronic treatment. It plays a crucial role in metabolic control and septal control through the actin cytoskeleton regulation. Long-term inhibition of mTOR is associated with a significant extension of lifespan. This slide represents the mTOR pathway and function relationship. You can see how mTOR acts. And in this slide, mTOR and its effect on autophagy is represented. Here how mTOR complex 1 effects on aging. You can see that mTOR complex 1 effects on stem cell mTOR complex 1 effect on stem cell turnover, cellular senses, inflammation, protein translations, and autophagy, and all these five factors are the responsible for the aging in the mammals. mTOR activation and inhibition. mTOR activation induces the premature aging of hematophytic stem cells (HSC). mTOR inhibition rescues hematophytic stem cells defects and extends life and hence extend lifespan. Rapamycin increased lifespan of mice. Research was done on the mice having the 22 months, and the results shows the animals which were given the rapamycin show more lifespan and less death ratio as compared to the other mice which were not given the rapamycin, and a significant difference was calculated among control group and the rapamycin treated group. Rapamycin, which is mTOR inhibitor, plays as a magic bullet role. It is used for the treatment of rejection in organ transplant, used in conjugation with coronary stents to prevent intimal hyperplasia after angioplasty. It is used in the treatment of hematological malignancies, used in the treatment of solid malignant tumors, also used in the treatment of tuberous sclerosis complex, which is a congenital disorder, and it is a potential treatment of autism, increased lifespan of mammals, anti-aging effect on adult stem cells. Conclusion: mTOR signaling is activated in aged HSC and causes decline in function. Inhibition of mTOR by rapamycin rescues HSC function in aged mammals. Inhibition of mTOR by rapamycin increases lifespan of mammals. Inhibition of mTOR stimulates immune function in aged mammals. Thank you.
Thank you, Muhammad Asim, for an interesting talk. And now it's turn for Muhammad Ramzan from Education University Lahore, and he will be talking about antibacterial potential of silk extracted from genus Argeo. Please, Muhammad Ramzan.
Bismillahirrahmanirrahim. Dear audience, I am thankful to the Applied Zoological Society of Pakistan for providing the platform of presentation. I am Muhammad Ramzan, MSc graduated, Department of Zoology, University of Education, Lower Mall Campus, Lahore. I am going to present about microbiology, and the topic is antibacterial potential of silk extracted from genus Argeo. I acknowledge the efforts and contributions from Dr. Saida Naseem and Professor Dr. Muhammad Arshad from University of Education, Lower Mall Campus, Lahore, and Dr. Hafiz Muhammad Tahir from Government College, University, Lahore. Actually, health issues and food spoilage are major problems of the world due to the bacteria. And WHO has described 12 different families of multi-drug resistance bacteria against which new antibiotics are urgently needed. About 25% annual loss of food throughout the world was recorded due to microbes, and important and improper and misuse of broad-spectrum antibiotics cause multi-drug resistance in many bacterial species. The antibiotics discovery was remained so rapid in 20th century, but the progress slowed down later on. Heimer gave the first report about the antimicrobial nature of spider silk in 1988, after which the number of discoveries have been made in this field. Spider silk has antibiotic potential, and it is biofriendly, and it is ineffective against mammalian cells and no autoimmune response in mammals. In 2011, Wright discovered that antibiotic agents in the spider silk are protein in nature. So the objectives of the present research work was to investigate the antibacterial nature of silk from spiders Argeo, and to detect that which solvent is more suitable to dissolve this silk. So the test units to detect this antibacterial behavior of the silk were Escherichia coli, Staphylococcus aureus, Pseudomonas aeruginosa, and Klebsiella pneumoniae. And the treatments were the two types of silk from Argeo spiders. The first one was web silk, and the second one was egg silk. We used 6 different solvents to dissolve these silks from the literature review. These were acetone, 5% sodium hydroxide, ethanol, methanol, distilled water, and urea solution. This urea solution was actually 1:1 mixture of two different solutions that you are looking on the slide. The positive control was tetracycline 30 microgram, and negative control was the respective solvents used for the safe dilution. This is a specimen from genus Argyop at Jallow Park, Lahore. So we collect the samples from Jallow Park and Changamanga Forest, southwest of Lahore. We also collect the web of these spiders in clean and uncontaminated F&F tubes, but the egg silk was only harvested in laboratory. Spiders were reared in large clean wooden boxes by providing houseflies as food and humidity and appropriate temperature. Silk was collected from these boxes with the help of sterile rods, and we prepared 3.5% solution of the egg silk and web silk in the described 6 different solvents. Then we heat these solutions at 47 degrees centigrade for an hour and then centrifuge at 7000 rpm for 30 minutes, and resulting supernatant were used as treatments. We prepared the bacteria cultures on nutrient agar media on the plates, and this diffusion method was used for recording the results. 10 microliter liquids from the treatments and negative control was poured on the prepared filter paper discs on the bacteria cultures, and prepared discs of tetracycline 30 microgram was used as positive control. Cultures were incubated at 37 degrees centigrade for 24 hours, after which diameters of zones of inhibition were measured as results. SPSS was used for statistical analysis by using MANOVA followed by Tukey's test, and the level of significance was 0.05%. Results for Escherichia coli revealed that the zones of inhibition for positive control was largest and have statistical differences with the negative control and treatments. But the zones of inhibitions of treatments was too small, and these zones were due to the solvents in which these silk were dissolved, as seen through the negative control. If we compare treatments with negative control, it reveals that the effect of treatments were due to the solvents that we use also in the negative control. And these are the results for all the 6 solvents in which the silk was dissolved: ethanol, urea, acetone, and 5% NaOH in distilled water. These were also the results for Staphylococcus aureus, and same results for Pseudomonas aeruginosa, and same results for Klebsiella pneumoniae. So we concluded from the result from these results that the silk from Argeo spiders have no active ingredient to inhibit the growth of these bacteria: Staphylococcus aureus, Escherichia coli, Pseudomonas aeruginosa, Klebsiella pneumoniae. But at the same time, we observed that 5% NaOH was more effective to dissolve the silk, and we also make further dilutions: 1.5%, 1%, and 0.5% for the web silk in 5% NaOH, and we make these treatments against the 4 bacterial species that I have already described, and the results were the same. So we recommend that actually a lot of spider species are discovered with silk that have antibiotic nature. These are some citations for in this work. So a lot of work is needed to evaluate the antibiotic nature of silk from different species of spiders which are still untouched. Further identification and separation of active ingredient in this silk need much work. The production of these biofriendly antibiotics at commercial level may also be possible through genetic engineering and other disciplines. So here are some references. One can follow these references in the citations given in the presentation. And that's all. Thank you very much. Allah Nigeban.
Thank you, Mohammad Ramzan, for sharing your research with us. And now it's time for Razia Noreen from Government College University, Faisalabad. And she will be talking about effect of Moringa oleifera leaves against phenylhydrazine hydrochloride-induced anemia in albino rats. Please, Razia Noreen.
Assalamu alaikum, dear all organizers and participants in International Conference on Applied Zoology. My name is Dr. Razia Noreen, Associate Professor from the Department of Biochemistry, And today I will present my topic of project, which is effect of the Moringa oleifera leaves against phenylhydrazine hydrochloride induced anemia in albino rats. First of all, the introduction about the Moringa. As we all are very familiar with this name, and we know this it has a lot of nutritional values in sense of multivitamins, irons, minerals, which are present in constituent amounts in this plant. Anemia is an iron deficiency problem, and according to WHO, anemia it affected the several peoples globally, and it is most common in Pakistan, especially in the pregnant women, lactating mothers, girls, and children. So iron supplements are required to cure this anemia, which again have some free radicals which affect the liver, because we are aware that all these kind of medicines, they have some side effects. So they produce the free radicals, and these free radicals indirectly affect the liver and other body organs. So Moringa, it has been reported as a rich source of iron and can be effectively used against the anemia and iron deficiency due to the presence of rich contents of iron in it. The objective of this study was a determination of iron status of Moringa oleifera leaves, and then the protective role of Moringa leaves against anemia. In the methodology, first of all, the Moringa leaves extract was prepared by maceration technique using 70% ethanol, and then iron content was determined by wet digestion method using atomic absorption, and anemia was induced in the albino rats by phenylhydrazine hydrochloride injections calculated according to the body weight of the rats for 2 days. Then we have divided our groups of rats. Albino rats were divided into different groups, and this study was continued for 21 days. Group 1 was a normal group which received the normal tap water and the diet. Group 2 was the negative control effect, which is after the induction of anemia rats with only tap water and the typical chow. Group 3 was after the induction of anemia rats, which were treated with the ferrous fumarate. And group 4 and 5, they were received Moringa leaves extract at 2 different doses according to the calculation of per kg body weight of the rats. So biochemical analysis for the assessment of induced anemia and Moringa oleifera leaves, we have done the hematological parameters, liver function test, and serum iron status was also analyzed. In the present study, the results showed that among different varieties, the Faisalabad variety has the highest iron content showed in their leaves, and phenylhydrazine toxicity produced the oxidative stress that affected the body weight of the rats. Hematological parameters were also affected. Liver and serum iron status was also disturbed by inducing this phenylhydrazine. So Moringa oleifera as a treatment, in the treatment groups, they showed that it normalizes the weight gain, it improved the hematological parameters, immunological response, liver functioning, and concentration of ferritin, transferrin, and total iron was also improved by giving this Moringa oleifera leaf extract. So Moringa, it acts as a hematoprotective agent and a hepatoprotective component for these albino rats. The results showed that the moringa leaves extract is beneficial than anemic supplements that have many harmful side effects, and it can be used for the iron deficiency problems in
evaluation of antimicrobial properties in fish skin mucus extracts of major cars. Fish, diverse group of animals. First of all, I will speak about introduction. Fish is a diverse group of animals, highly specialized for their aquatic existence and comprising almost half of the number of vertebrate species. Globally, fish provides about 3% of annual protein. Global fish production was 171 million tons in 2016. Pakistan fisheries contributes about 1% to its GDP. Per capita fish consumption was about 1.9 kilograms annually. Fish coexist with many of pathogenic and non-pathogenic microorganisms and have complex defense systems which ensure fish survival. Infectious diseases cause multi-million dollar loss annually and reduce the economic income. It causes mortality in fish. Innate defense system helps a fish to maintain healthy state under normal conditions. Skin mucus is an important component of innate immune system and acts as a barrier between fish and the external environment and plays a frontier role in fish production. Continuously get replaced and prevent colonization of bacteria and fungi. It also contains a number of factors of innate immunity like proteins, enzymes such as lysozymes, immunoglobulins, complement proteins, lectins, C-reactive proteins, phosphatic enzymes, alkaline phosphatase, and other components of antibacterial proteins and peptides. Fish skin mucus exhibits significant and specific antimicrobial, antifungal, and cytotoxic activity. Composition and rate of mucus secretions varies from species to species and in response to microbial exposure, environmental fluctuations, etc. Fish plays an important role in respiration, ionic and osmotic regulation, reproduction, excretion, disease resistance, communication, and protection. Potentially makes mucus a highly multifunctional material. Let's move towards materials and methods. First of all, experimental fish, Libya Hydrovitas and Highness Caligula and Ketla were purchased from government fish hatchery, Tiana Road, Hyderabad, 250 to 300 grams in weight. Fish was stocked in glass aquariums having water capacity of 36 liters. Then fish were acclimatized to the laboratory conditions for 15 weeks protein diet. After acclimatization, for 10 days, fish were used for the mucus collection. Before mucus collection, fish was starved for 24 hours and was placed on a specimen tray. Mucus secreted on the surface of body was collected as a sample with the help of sterilized spatula and was stored in sterilized apron drums. Then mucus was centrifuged at 5,000 RPM for 15 minutes and stored at 4 degrees centigrade. Then acidic mucus extract was prepared. For acidic mucus extraction, 1 ml of fish mucus was mixed with 0.5 ml of 3% acetic acid, which obtained concentration of 2:1. 2% was fish mucus and 1% was 3% acetic acid. Then apron drops were placed in boiling water bath for 5 minutes and then was cooled in ice and centrifuged at 12,000 RPM for 35 minutes. Antimicrobial assay, while the previous method was used to determine the growth of inhibition effect of skin mucus against aggressive bacteria and fungi. Let's move towards the results. Zone of inhibition of gram-negative bacteria, Aeruginosa hydrophila against Aeruginosa hydrophila by Libya Highness Caligula, Ketla, and control was specific time. Antibacterial drug. The highest zone of inhibition was observed by the Libya which was 20 mm in diameter, and Highness Caligula comes at second position, 16 mm zone of inhibition, and Ketla with 13 mm diameter of the zone of inhibition, and the control drug with 25 mm diameter of the zone of inhibition. Then at second number, gram-negative bacteria is the zone of the graph representing the zone of inhibition of all the fish mucus extracts, Libya and Highness Caligula, and control drug. In the against the Staphylococcus aureus, highest antimicrobial activity was observed by the Libya, then Highness Caligula and Ketla. The control drug shows the zone of inhibition of 25 mm, radial meter 23 mm, and Serhinus midland, the Ketla, with the diameter of zone of inhibition of 21 mm in diameter. Zone of inhibition, antifungal activity of three carp species against the flagellant axis form. The highest antimicrobial activity was shown by the Serhinus midland with the with diameter of the zone of inhibition 19 mm. Then radial least activity was shown by the Tekla. Zone of inhibition of fungal species as per genus Niger by radial Vita, Serhinus nigla and Tekla. The highest antifungal activity was shown by the radial Vita, which was 17 mm in diameter and 15 mm in diameter of the zone of inhibition was by the Serhinus nigla, and 13 mm diameter of the zone of inhibition was Ketla, and 25 mm diameter of the zone of inhibition was the control drug, which was gentamicin as an antifungal drug. Analysis of all the fish mucus extract against all the tested bacterial and fungal strains. Blue Dracosaurus, pink, green, and each color representing a different bacterial or fungal strain. And fish species. As the graph representing the highest antimicrobial activity was represented by the nucleotida against the S. aureus, which was 23 mm in diameter. And at the controls are also representing the diameter of zone of inhibition. The conclusion, the findings of the present experimental trial suggested that the acidic mucus extract of all fish exhibited the antimicrobial properties against the selected bacterial and fungal strains, which might play an important role in fish defense against the pathogens. Thus, it appears to the possible use of steam mucus of carps as a potential source of antimicrobial agents in alternative to antibiotics against the fish and might be in human infections. References and thanks for your attention.
Thank you for your nice presentation. And now it's turn for from University of Agriculture, Faisalabad, and which is entitled Determination of Antimicrobial Activity of Date Palm Pits Against Food Spoilage Bacteria. Please, Afshin Mahmood.
Myself, Afshin Mahmood. I done my BS honor in microbiology from the University of Agriculture. I also done my master degree MPhil microbiology from same university. Now the topic of my concern is determination of antimicrobial activity of date palm pits against food spoilage bacteria. Here is the introduction. Decaying of food spoilage bacteria, approximately for damage of worldwide food spoilage due to microorganisms. Worldwide microorganisms destroy the different types of food. Pseudomonas are renowned as major food spoilers. Gram-positive bacteria like Staphylococcus aureus are the main cause of food spoilage. Members of gram-negative bacteria such as E. coli also take an active part. 88% of different varieties of date having a bits of Phoenix dactylifera containing several important elements such as alkali, soluble polysaccharide, ethanol, and citric acid. It is considered as a part of Arabian diet and also a low cost of food that is cultivated for eatable sweet fruit. Next one, date palm pit as antimicrobial includes antibiotics in addition to other chemicals and compounds that are used to kill microorganisms. Antibiotic resistance using plant extract to act against food spoilage bacteria as it have phenolic compound like flavonoids and phenolic acid. Antimicrobial agents reduce the growth of microorganisms, credit largely to their carcinopreventive and antimicrobial agents. Here is the objectives of my research topic: extraction of date palm pits and evaluation the antimicrobial effect of date palm pits against food spoilage bacteria. Material and methods. Date palm samples were purchased from local market. Then samples were transported to the research laboratory of microbiology, were removed and dried. After dried, we using mechanical grinder. Uh, uh, this grinder were used for then ethanolic solvent of seed powder were extracted by using the social extractor. Here is the four bacteria which was used: E. coli, Salmonella, Pseudomonas, and Staph aureus, which were used in procured form. And then after this, I perform antimicrobial screening. Agar diffusion method were used to check the antimicrobial screening of the ethanol extract of the date palm bit. Subculture plates were incubated for 24 hours. Sterile cotton swab were used to perform lawn culture. Sterile cork borer were dipped into the agar to make holes. Each correct antibiotic discs were used as positive control. The zone of inhibition was checked. Here is the results. Identification of bacteria isolate confirmed through gram staining and biochemical tests. The phytochemical screening of date palm Phoenix dactylifera seeds with extract shows that alkaloids saponins were present in ethanolic seed extract of Phoenix dactylifera, while steroid and tannins were absent in pit extract. The initial weight of seed sample were 30 gram while the weight of extract was 9.2, thus the percentage of yield was 42.8%. Depending upon the obtained result of antimicrobial activity for the pit extracts of the date, the test performed in test tubes for the estimation of antimicrobial efficacy of antibiotic ciprofloxacin, and also the pit extract was investigated against Escherichia coli, Pseudomonas, aeruginosa, Staphylococcus aureus, as well as Salmonella. Antimicrobial effect was revealed by way of the inhibition zones of the mean diameter. The seed extract was found effective at 1000 microgram per milliliter as against Escherichia coli and Pseudomonas, as it demonstrated higher zone of inhibition, that is 20.4 millimeter and 15 millimeter, while the pit extract found to be less effective at 1000 microgram per milliliter as against Staph aureus and Salmonella, as it demonstrated lower zone of inhibitions, that is 9.5 millimeter and 11.5 millimeter. Here is the pics which shows the zone of inhibitions. First two pics in which the ethanolic date palm extract sensitivity against E. coli and Staph aureus, and the next one ethanolic extract against Pseudomonas aeruginosa, and the next is Salmonella typhi. Conclusion: the present study indicated that the prevention of bacterial growth by Phoenix dactylifera pits ethanolic extract could be due to the existence of few active phytochemical compounds which may take action by oneself or in combination to prevent the growth of food-spoilage bacteria such as Escherichia coli, Pseudomonas, and Salmonella. Thank you.
Thank you, Afshin Mahmood, for your nice presentation. And now it's time to move towards our last speaker of this session, Sabir Hussain from Government College University, Faisalabad, and he will be talking about biodegradation of Isoproturon by a soil bacterium, Syngiobium species S29 in soil and Equipedia. Please, Sabir Hussain.
Bismillahirrahmanirrahim. Dear fellows, Assalamu alaikum. I am Dr. Sabir Hussain from the Department of Environmental Sciences and Engineering, Government College University, Faisalabad. Today I am here for an oral presentation in the third International Conference on Applied Zoology. The topic of my presentation is biodegradation of Isoproturon by a soil bacterium, Sphingobium species S29 in soil and aqueous media. As you know that pesticides are used in conventional agriculture to ensure the quality and quantity of agricultural yield. These pesticides might be in the form of fungicides, herbicides, and insecticides. Although the pesticides are used for the improvement of agricultural yield, but at the same time, they are source of pollution of the environment. Isoproturon is one of the phenylurea herbicides which has been commercialized for more than 50 years. It is used for pre and post-emergence control of broadleaf weeds in cotton, fruit, cereal cultures, and other agricultural crops. It is a systemic herbicide which inhibits the photosynthesis of the target weeds. Ecotoxicological data suggest that it is toxic for some invertebrates. It is suspected to be carcinogenic and genotoxic. It causes reproductive and developmental toxicity in human beings. In addition to this, it has been detected in waters in several countries, including Pakistan. After entry into the soil, few previous studies have reported for a very low mineralization of this herbicide in the soils. However, during the recent years, some of the studies have reported for enhanced degradation of this herbicide in the soils repeatedly treated with this herbicide. This phenomena is attributed towards the adaptation of microorganisms for isoprotein degradation. The microorganisms use this isoprotein as source of their energy. This phenomena has been observed in different countries including UK, Denmark, France, China, and Pakistan. On the basis of these results, several researchers have isolated different bacterial and fungal strains involved in the biodegradation of isoprotein. Here are a few strains of bacteria and fungi which have been isolated and characterized for biodegradation of protein. These are the strains which have the potential for mineralization of this herbicide. In this context, the present study has been planned with the objective of isolation and characterization of a novel bacterial strain involved in isoprotein degradation in aqueous as well as soil media. For this bacterial strain was isolated through an enrichment culture technique in the minosalt medium using isoprotein as only source of carbon. This strain was identified through PCR amplification, sequencing, and bioinformatic analysis of its 16S ribosomal DNA gene. The potential of the strain S29 for degradation of isoprotein as well as its known metabolites was tested in minosalt medium. The potential of this strain to degrade other phenylurea herbicides was also tested in minosalt medium, and the capabilities of this strain to degrade isoprotein in the mineral salt medium was also tested at different temperatures and pH values. And finally, this strain was tested for degradation of isoprotein in the soil. It was found that this strain had a good potential for degradation of isoprotein as well as its known metabolites as shown in this figure. On the basis of this finding, it was suggested that this strain followed a degradation pathway in which the isoprotein was demethylated twice and then ultimately it was transferred into 4-isopropyl aniline. This pathway has already been reported for some other bacterial strains belonging to genus Sphingomonas. This strain had the potential for only further degradation of isoprotein, and none of the other members of phenylurea herbicide was found to be degraded by this strain. It means that this strain has the enzymatic system which is highly specific for degradation of isoproteron. The optimum temperature for degradation of isoproteron by this strain was observed to be 30 degrees centigrade, and on other temperatures, the potential was found to be reduced. The optimum pH for isoproteron degradation by this strain was found to be 7, whereas it was decreasing with any increase or decrease in the pH values. And furthermore, it was observed that degradation at different pH values is also associated with the growth of the strain at different pH values. In the soil, it was found to have a good potential for degradation of isoproteron. This study indicated that in the soils, by augmented with the strain S21, S29, whole of the isoproteron was degraded only within 20 days. Hence, this strain has a good potential for the degradation of isoproteron in aqueous as well as soil media. It can be concluded that this strain might be used as a potential candidate for the bioremediation of the soils as well as aqueous media contaminated with herbicide isoproteron. Thank you very much for your attention.
Thank you, Sabir Hussain, for your nice presentation. And with this talk, our today's second session has come to an end. Now, our next session will be moderated by Ms. Rabia Zara, and the session will include talks from the fields of parasitology, entomology, toxicology, and pathology. Stay connected with us.